My goal today is to review some concepts of cerebral excitability, primarily the excitability of the cerebral cortex, and to show how these are related to physiological parameters, including waking and sleep. The idea is that during waking, there are progressive and cumulative changes in the cortex that lead to a need for sleep, a homeostatic regulation for sleep. And there's some exciting new findings, as well as some earlier findings, suggesting that these are measurable in the EEG. So let me first be sure that you can hear me. Is the audio okay on this?
Yes, sounds good.
Yeah, good. Okay, so this is a kind of work in progress. Basically, it's some new reading I've been doing and trying to relate it to previous work that we've done. And so this should be informal. So I would say break in at any time and we can discuss this and try to make sense of the data I'm going to present and some of the implications. So we're going to look at activation of the brain, primarily the cortex, the cerebral cortex, and trying to think about the underlying physiology of it. We can see the brain waves change. For example, we can see slowing with decreased arousal, and we can see kind of a desynchronization pattern under high arousal. And what are these showing us about the brain? And there's a concept of excitatory inhibitory balance that's somewhat complex, but an interesting one to those who are modeling the brain in relationship to physical properties or physical processes, where critical states may be reflected by this excitatory inhibitory balance. And I'll try to explain what that means. But practically, the restorative function of sleep is a mystery. And some of the data I'm going to show suggests that the normalization of the process of waking, this activation process of waking, happens in sleep, and it determines the restorative function, what makes a good night's sleep. And so understanding this measure is going to be very important, both to our goals to develop high-density EEG and to our efforts to do sleep therapy, which involved kind of a dynamic, ongoing sleep analysis. Now, some of the concepts will be familiar from our previous workshops and seminars in terms of understanding how the EEG is generated from neuronal activity, the activity of neurons in the cortex. And some of our first seminars, Fan Lu led, on the models of the cortex. So we know if we go back to the basics of EEG, it's generated by excitatory and inhibitory changes within the cerebral cortex, and their balance will create these functional dipoles, basically transition between negative and positive fields created by neurons, not just their firing, but by their graded and some synaptic potentials. Now, those have to be coordinated in order to make a field at the EEG electrode, but they seem to be reflective of the cortical columns, groups of neurons that are in a package, basically across the 6 layers of the cortex. And understanding the circuits of those neurons in each of the cortical columns might give us clues to how together, those columns will generate the EEG. And so this is some of the beginning work that Fawn was exploring with the cortical columns and the canonical models of cortical microcircuits. And of course, we are interested in that as we're mapping them with our source analysis techniques. That basically, we're searching for those dipoles that we could measure at a far field and understanding the local circuit dynamics that regulate it. So the work I'm going to describe today comes from Lender and Associates, where they have some new measures of cortical excitability that they start with in mouse brains where they can measure calcium, and then they relate to the EEG features, both from the mouth, but also then in humans. And what they find seems to be evidence that sleep, particularly in this case, REM sleep, does something to these synaptic strengths to decrease the progressive excitability that happens during the day, and therefore returns the brain to some homeostatic state. So we're going to try to decompose that and relate that to theories of sleep, like the Thorelli and Tononi synaptic renormalization hypothesis, that basically says sleep does something to undo the excitability of the synapses that build during the day. And so we're going to look at some of the measures that are derived. So here's the paper. It's fresh out in Science Advances. And we recognize a lot of the names here as people who've been very active in sleep. And so we're interested to understand this effect and what the nature of the data are. So as I mentioned, there are two kinds of data here. And again, stop me if something's not clear, because I'm still trying to understand this and may not be so clear in explaining it. The one kind of data is calcium imaging, a very cool new technique, where basically they shave the mouse's skull so it's thin enough you can look through it. And when you look through it with labeling of calcium, you can see specific neurons. So here they're looking at pyramidal neurons, PYR, parvocellular interneurons, a certain kind of neuromodulator of some of the inhibitory interneurons, and the somatostatin interneurons as well. So it's kind of a cool technique that they can look at each one. They look at the different stages of sleep and the calcium activity in each of these neurons gives a clue to its excitement or activity level. So we'll look at the progression then across this, and what we see is kind of the two striking features here. So for the pyramidal neurons, wake seems to have the highest activity level, and you can see the distributions across that area for waking. The non-REM has less, and REM has the lowest pyramidal activity, although look at the spread across REM. That's kind of interesting. And then the other marked difference is the parvocellular interneurons, where their activity is high in REM. So you might think, and of course it's highly variable. You see the bell curve or the distribution of activity also shows high variability in REM. So you can see that REM seems to have a high degree of inhibition as measured by the calcium imaging. Now, they also then measure the EEG in the mouse at the same time. And here's where the power spectrum becomes the measure of interest. The power spectrum is the distribution of activity across frequencies. So on the lower left-hand chart, you see the frequency resolved from 10 to 30 hertz here. And higher than 30 hertz is important as we'll see. And there are two components of the power spectrum. One is the more typical component, periodic. So delta, theta, alpha are periodic or oscillatory features. But there are also non-periodic features that can be extracted from the power spectrum. So then you don't see bumps at each frequency of interest like delta. What you see is a flat line. It's basically a 1/f distribution. In other words, the reciprocal of frequency. And the measure of interest for us will be the slope of that aperiodic spectrum. And basically what it means is during higher activation, higher excitement levels, that'll be a flat slope. Basically what that shows is the high frequencies are enhanced. And for lower activity, the lower frequencies are enhanced. Again, all aperiodic, in other words, they're not cycling or oscillatory. They're more intrinsic, maybe. So then we see that slope of the power spectrum. Again, a flatter slope or a lower number means higher frequency activity and more active. So the lower right plot now is showing us that during wake is the highest activity level by the electrophysiologic measure. During non-REM is actually lowest, and REM is somewhat higher. So there's something of a divergence between the calcium measures and the EEG activity measures, which is important to appreciate. And yet, nonetheless, there seems to be some association of each measure of activity or indication. So in these chart,
Don, can I ask a question on the previous slide?
You bet. Yeah.
So the C right, this is the also EEG measure.
That's right.
So this is the EEG. So they're still not relating this cell type to this specific activity. Okay.
Not yet. That's right. Well, and we can't exactly. In other words, the calcium differentiates the cell type, but the EEG, we just see the field of all cell types.
No. My question is if they're coming to this correlation and they see if, for example, pyramidal cells are more correlated with this EEG activity.
Okay. Well, we will get some clues to that.
Mm-hmm.
I think in this next slide here. Although it gets a little confusing when we get to this excitatory inhibitory ratio. I think I know what they did here, but it's a little confusing. This paper.
Backing up a little bit, Don, to your previous slide, the EEG technically only reflects the pyramidal activity, and and indirectly the inhibitory, meaning that they modulate the activity of the pyramidal cells, but the EEG pick up the pyramidal cell activity because of expansion. Right. But here, so both PV, parvalbumin, and somatostatin are inhibitory, right?
That's right. Well, no, somatostatin is not in this case, and we'll see that it's really the parvalbumin that has the big inhibitory effect in these data, anyway.
Oh, okay. I thought somatostatin would be a type of inhibitory.
Well, it's an interneuron, so I would assume that as well. But
weird. Okay,
it's a little paradoxical, and you can see here it doesn't have that big parvalbumin change in REM.
Right.
So I'm not sure I can explain that.
But then, so EEG here on your aperiodic activity and the slope they take it from just the slope starting at the lowest frequency to the highest frequency.
Yes, but I think they've extracted the periodic component.
Right,
so
right.
This
so
is
the inset, that little inset that
Right.
says aperiodic. So it's they're calculating the slope of that.
That's correct. That's
Okay.
correct. And you'll see that in several slides. That's their metric.
Yeah,
And
I
it's
see.
when it's
I
yeah,
see
the
why
flat slope.
their slope measure is essentially the same for wake and REM,
Right.
because the curve
That's
is
right.
okay. Yeah, yeah.
Now, the distribution's different, which I think is important, but
Yeah.
that's right. The slope is their measure, and they relate that to excitatory inhibitory balance. And this is a little tricky, but interesting.
Don,
Let's
I'm
see if I
slightly
can.
So consider that somatostatin interneurons can disinhibit PV, parvalbumin neurons.
Yeah.
So it can get kind of tricky in the interpretation.
Well, that's good. That's good to know, to try to help interpret it. The somatostatin doesn't come out so interesting in these analyses.
Okay.
All right, let me go on and see if I can sort through this. This is one of the harder things to explain, because what they're trying to get to is the excitatory inhibitory balance and ratio. And it's a little fuzzy reasoning that they hand wave about. But let me see if I can sort through these. So now they're relating the EEG activity, particularly these slope measures, to the excitatory pyramidal activity. And of course, that is the pyramids neurons are the big generators of the dipoles. Although to the extent that the inhibitory neurons modulate them, that changes the EEG equally well as the
Exactly.
activity of the parameter neurons themselves. So, and somewhat, this is why the calcium imaging is pretty cool, because you get to see the individual contributors to it. But you see here, if we look at that PSD slope, power spectral density slope, on the upper right figure there, you see a pretty straight correlation, although the distribution runs around there. For the quartiles, the bottom quarter, the middle 2 quarters, and the top quarter, you see a pretty tight relationship to the flatness of the slope. So that does say that the more pyramidal activity indexed by calcium, which is an indirect measure, goes with greater excitability by the flatness. In the upper left, you see the power spectrum flatness crossing over at 30 hertz. So the flatter power spectrum is the so-called more active feature. And we're going to see that gets flatter during the day as you have sleep deprivation, or even over multiple days of sleep deprivation.
Oh, so what they did here was they looked at the calcium activity and divided it into quartiles of how much activity it represents. Is that right?
That's right. They just look at the distribution, right?
Yeah, and then they use the calcium activity quartiles to sort the slope, the EEG slope.
That's right.
That's
okay.
Right. Okay.
And so we see you get both measures, and this shows how they're correlated significantly.
Yeah. Okay.
And then in the bottom slide, they measure the excitatory to inhibitory ratio. And I've gone through this paper to try to be sure about it, and I'm mostly sure that they're actually measuring the inhibitory neurons and the pyramidal cells to get that E to I ratio. So that's the typical neuroscience pyramidal to inhibitory or pyramidal to interneuron showing the excitatory to inhibitory ratio. Now that's kind of key because that's going to become functionally important to the issues that we'll raise later. So you can imagine that neural activity, you could increase not just excitability, but the inhibitory counterpoint. And that will become important to the function of the cortex, as we'll see in a few minutes. But basically, when they relate the aperiodic EEG, you get kind of a similar function where it becomes flatter with higher activation. And so, although as I'm reading that lower left-hand one, the fourth quartile, the high excitation, is showing a steeper function. Whereas
the power
the more
AU,
inhibition.
power AU is area under, or what?
Yeah.
What is?
It's the units of power of the power spectrum.
Oh, that's mu, microvolt, or it's not.
No, I think the AU is the unit of power.
Okay. Is it arbitrary unit? I think that's what it is. That's what probably AU. Okay. So let me just see this. Okay.
But here you see that the excitatory inhibitory quartiles don't have that tight relationship to the slope. But then they try to put that together with those EI quartiles against the pyramidal activity over on the right. And I think what they're trying to show is the higher pyramidal activity from the calcium measure is different from the EI quartiles, because you see as the EI quartiles go up, that highest level of 4 only has the high pyramidal activity for the upper right-hand quadrant. So whereas you can relate the pyramidal activity to the excitability measure from the EEG, it's not so clear for the excitatory to inhibitory balance. Okay. What happened to my okay. So the in this paper, not only did they have the rodent, but they also have the human data, and where they're relating this kind of restorative function of sleep to a change in the power spectrum slope. And so over the left-hand side, what they have is what happens during sleep. So between the PM, the night before sleep, when you see the day's activity is causing a greater flattening of the power spectral density slope. So you see how the blue line is flatter. In other words, it has more high frequency activity in the power spectrum. And I think this is the total power spectrum here, because we have more bumps on it showing the oscillatory activity. But then the next morning, that has been changed. That the red line is showing a decrease across those higher frequencies. So that becomes the most interesting measure, I think, of sleep renormalization of that high frequency activity, regardless of how we relate it to pyramidal and inhibitory neurons or the excitatory inhibitory balance. Clearly, there's a change in sleep that would support a renormalization kind of hypothesis, although exactly what that is still is unclear. And then on the right, they relate this to memory performance with fairly impressive result. that the memory retention is associated with a change in that slope. So the degree of that slope normalization goes with better memory the next day. So that suggests that it's not just a normalization of the EEG, but it has functional significance for memory consolidation. Although how that happens is kind of not explained very well at all. So these next slides will show the sleep deprivation effects, various ways of changing the power spectral density slope with the lack of sleep, or the renormalization that happens in sleep. So up on the left, we see either habitual sleep, so you had a good night's sleep the night before, or at least your typical one, or sleep deprivation, you got no sleep. And the sleep deprivation looks like the all-day activity. So the slope is flatter, mainly because the higher frequencies, not synchronous frequencies, but broadband, are increased. And that's shown on the upper right in terms of an increase in that distribution after sleep deprivation for that general PSD slope. And then another plot, the lower left, they simply show the night before what your slope is in blue. You can see the high frequency activity. And after sleep, that drops down. I think for our purposes, it's kind of a cool metric because we could show the efficacy of any sleep therapy that we do by a measure like this, a simple measure that we already get in the EEG.
Is this done across all EEG channels?
They specified the channels up in the inset there. Yes, it's done. This is kind of an average across all of the channels they used.
Okay.
But look at that plot there. The significance of that difference is particularly big around the midline.
Yeah.
Almost like,
I
don't know what exactly that means. That kind of midline distribution there, there's only two things we see that. One could be spindles, but the other is that REM oscillations, like from the Silicari and Tononi publication. So there is some regional distribution that isn't shown very well in this paper, but it's interesting. Okay, and then finally, this slide's somewhat interesting, and I don't want to spend too much time on it because I do want to go on. So here is the broadband spectrum at the top for the non-REM before a REM episode and the non-REM after a REM episode. They just selected the non-REMs around the M to look at the before and after. And then the oscillatory residuals, this is after you take out that aperiodic slope and look at the features that are oscillatory. Now, I find this interesting because what it shows is big theta activity during REM. And humans aren't supposed to have that. There's an argument, at least you see repeated a lot in the literature, is that whereas rodents and most other mammals have big theta and REM, humans do not. And that's clearly not the case in their data. And I think maybe it's because the aperiodic features are so big in human EEG, we miss the theta oscillation. All right, so I want to go on to other ideas about this excitatory inhibitory balance that may give us insight into kind of the neural significance. So when we say that this balance is being changed during sleep, What does that mean? How could it relate to the function of the cortex? I mean, I think the synaptic renormalization idea sounds kind of good, but what does that mean? How does the excitability of the cortex relate to its functional capacity? And here is where some of the excitatory inhibitory balance research has been really quite interesting. And I think it's worth looking at to give us an idea of what these EEG changes mean. And this will take us back into some of the canonical cortical microcircuits models, where the excitatory inhibitory balance seems to be important to the function of neurotransmission, not just within the column, but across columns. And on the right, we see some of the measures of neural connectivity where the transmission of charge and presumably information between neurons doesn't happen at a subcritical phase, when the excitatory inhibitory balance isn't up to a critical stage. Now we know that if excitation gets too much, then we have seizures. And not only do we see that in neuroscience models, including slice preparation, but in neural network models. When the excitation goes beyond the inhibitory control, there's runaway excitation. So the idea of criticality is the excitatory inhibitory balance is at a high stage, but a balanced stage. And this relates to the physical model of criticality, physical systems that maintain themselves in a phase state below runaway distribution or changes. And so I'm going to talk a little bit about self-organized criticality and its operation in neural circuits, and give some specific examples that suggest that the excitatory inhibitory balance is essential to function, that transmission of neural activity across an actual cerebral cortex network depends upon being near that criticality. And beyond that, there's evidence that the mammalian cortex maintains itself at that critical state as an essential physiologic self-regulation. And before I go to that, let me just point out this feature of critical states in physical systems. And this physicists like this kind of analogy because it suggests the balance of regulatory systems, and it's been applied to neural networks, is what I'm going to show next. So here's the original model. We have the sand pile, and a sand pile is maintained at a level of criticality where the gravitational effect and the friction are in a reasonable balance. So as you add more sand grains in a sand pile, you get up to a critical slope. And beyond that critical slope, you get avalanches. And that's where one grain of sand can trigger a whole bunch of other grains of sand to have an avalanche, to fall down. It's the same thing with snow. And the equations that describe this show that that criticality is a particularly important functional state where things are in exact balance. And in fact, the avalanches are used to describe the spread of activation in cerebral networks, things that will be important as we're trying to understand activity during the day and how that reaches a state that's unstable until you have sleep and restore the network back to the stable dynamic state. So this is work that Dietmar Plenz has done over a number of years. This paper is a recent summary of it. And it's really interesting stuff. But basically, this will show that the cortex maintains itself in a state of criticality, where the excitatory inhibitory balance is very close. You're kind of right under the ratio of 1, which means that the inhibition, I think, dominates just slightly, so the excitation doesn't run away. And the technology is kind of cool. It's a different kind of EEG than we normally look at. What they do on the left-hand side, you see a little array of elevators, and on the right, and then you see the recording apparatus on the left-hand side of this incubator. And every one of those little trays on the right-hand side is a mouse infant, a kind of a newborn mouse pup, whose somatosensory cortex and part of the midbrain have been shaved off to create a slice preparation. So they can rotate those in, do individual measures on them with that device. The middle frame shows the electrode array superimposed over that cortical slice. And then what they do is they do this right at birth, and then they grow that slice preparation. So the mouse brain continues to grow, to self-organize, and on the right-hand side, it has brought in more inhibitory controls. Those become mature and form a relatively mature cortex. Now, there are a bunch of interesting things beyond this, including the ventral tegmental area with dopamine that is quite interesting in the regulation of the activation level of the multi-activity that's indexed there. But the main point of this is the way that slice maintains itself at criticality. So the criticality shows that the neural transmission, when the excitatory inhibitory balance is right at the right stage, is highly effective. However, if it goes too much, then you have seizures that propagate of this, too much excitation, and it's not balanced by inhibition. So changes either in excitation or inhibition that take it away from that criticality will lead to dysfunction. Inadequate excitation leads to non-function. The neural transmission does not happen. And you can look at this, unit A on the left is a neural network model representation, a machine learning artificial neural network that can be set up to show this propagation of activity that's right at the criticality state. And if you're not at criticality, it won't propagate. And Plenz and his associates over the years, with their preparation of the newborn mouse brain, have been able to show that that this operates in the neural tissues. When they're at criticality, they show propagation through the network, and when they're below criticality, they don't function. Now, the most interesting thing is if you perturb this. So if you add an inhibitory neuromodulator to the slice preparation and put it out of criticality, it brings itself back to criticality as part of the normal developmental mechanism. And I think that's through the excitatory inhibitory balance, that the physiology itself will regulate it back to that again. So as I've been trying to read to understand this kind of dynamic regulation of excitation and inhibition, and as a clue to how to understand sleep effects, I came across this recent paper that I'm only now trying to get through, but it has very good evidence that long-range connections shown by long-range temporal correlations in humans measured with EEG will decline with sleep deprivation. So let me just go back on a couple of points. So this is done by Christian Meisel. And some of you will remember we worked with him with his ideas of phased enhancement, phase synchrony during waking that does decline during sleep. So it's another measure of this excitability that the regions of cortex become in phase with each other in the EEG. It's an alternative measure, but I think relevant. What they show in this paper fits with the idea that criticality is necessary for neural transmission, because what they show is that when you lose that high frequency component of the EEG because of extended waking, sleep deprivation, then the long range correlations decline. So it's as if you can't maintain that propagation of activity through the cortical tissue, then it can't maintain long-term relationships. So I'm still trying to understand this, but this will really be very relevant to our efforts to understand the regulation of activation of the cortex, including by motivational factors and the emotional arousal that's very critical. So here's the recent paper Fawn and I have been developing, trying to understand the way that the activity of the entire cerebral hemisphere is regulated by activation control from limbic regions. So my goal is going to be in future talks to come back to these basic mechanisms, but then extend it into the human brain and the organization of excitability and activation level as a function of particular sleep mechanisms. All right, so I'm going to stop there and see if we could review back and get the basic concepts to this point.
Pretty cool. It would be, I guess, you know, how replicable the EEG findings are in terms of those measures.
That's right. That's right. Well, and I think we're going to we could look at this with data we have right now.
I was going to say that. I think we should be able to do that. Do you know if they did this with a toolbox that we can have access to?
I'm pretty sure it's open. Yeah, I'm pretty sure the aperiodic and periodic techniques are available. I don't think there's anything secret about that.
Yeah, this is where if we had their exact workflow, I mean, you have the toolbox and all that, but you know how they clean their data, et cetera, and which stage of sleep. I guess this is all stages of. No, this is wake. Upon wake, PM is before sleep, and then
that's right.
AM is at. Okay, so this is all waking.
That's right. In the humans, all of the data in this paper is waking, EEG. Now, but you know, you can see in the mouse, they're doing the measures during the stages of sleep. But I think for us, one of the interesting questions is, what is the EEG frequency characteristics, particularly these aperiodic ones, during the stages of sleep?
Yeah. Well, and I'm also a little surprised here, you know, that they don't go into the high gamma or much into gamma, right? This slope ends at what, 50?
Right. Yeah, I agree. They do some of the measures with the animal Well, they don't show it. I think that's just their presentation method, because I do think, we've seen before that the aperiodic EEG up into high gamma, as you mentioned, is a very good index of local activation. Like with Kai Miller's recording during neurosurgery.
That's exactly what I was thinking about. The
Yeah.
broadband gamma. I mean, because
Exactly.
that's what you think would be the big effect, or I mean that's that, yeah. So yeah.
That's correct. And with our EEGs, we can get up there. We can certainly get up to 100 hertz, even with our 250 sampling. So we should be able to sample that pretty directly. But one of the studies I think would be very interesting is to measure the change from the nighttime to the morning time, basically, which is what they're looking at here, as a function of each stage of sleep. They argue, and I think it's somewhat arbitrary, that it's REM that's doing this big change. Well, we know REM is not so important to explicit memory. Here they have an effect from REM, but it looks much more like it's non-REM that predicts the explicit memory. And of course, that's what we're manipulating with slow wave sleep.
Right.
So to me, there's a whole host of questions that could be very relevant to our measures, where we're trying to say that sleep is relevant to daytime cognition and function. And that's why particularly, I just found this Meisel paper. I had it, and I hadn't read it very carefully. But if the cerebral networks are so disengaged, unless they're at the right criticality, that's really huge. I mean, so our whole theoretical paper that we just published in Cerebral Cortex is about the functional connectivity networks, the networks that are so popular in fMRI, the dorsal attention network, the ventral attention network, the default mode network, the salience network. I mean, we're trying to argue theoretically that these are regulated by limbic structures, but if we could get tight measures of the network dynamic, you could see how critical the sleep stages are to those differential networks.
Overall, pretty cool though.
So I think we should try to organize some of the analytic techniques with our current data. I think even the WISP data that we have on the replication of the Hathaway results would be a good first step to look at the extent to which changing in 3 sleep may change the waking nighttime to morning EEG differences. These are pretty straightforward metrics for us to develop.
Yeah.
Any other questions?
What is, in general, I don't understand the concept of aperiodicity. I know it's 1 over F, but what does it mean, difference between periodicity and aperiodicity?
So it's, if you imagine kind of stochastic processes, things that have no periodicity in time, but are still active, they will have frequency components. They're just somewhat random. So now in the EEG, Fon was taught, we see that. We see that in the the cortical EEG, particularly if you're measuring it directly from cortex. So in the motor studies that Kai Miller did at the University of Washington, he was a really good PhD student, but he eventually became a neurosurgeon because he could make a lot more money as a neurosurgeon. He's now at the Mayo Clinic. But what they did, this is in Jeff Ogeman's lab, and we followed this pretty carefully. What they showed was they measured the EEG directly from the cortex, from intracranial electrodes during a motor task, and they looked at the activity of different fingers. The changes that were most indicative were aperiodic. In other words, there was not a gamma frequency that showed the change. It was a whole range of frequencies, probably over the gamma interval, low and high gamma both. So it's aperiodic. In other words, there wasn't one frequency that popped up. It was the whole range of frequencies, mainly because you're probably summing over many gamma networks, each of which has a slightly different tuned frequency. So it's like a flat power spectrum. You know, no frequency has dominance, but because the basic function, lower power is bigger, that makes the 1/f distribution.
So how does the graphs in, I think, the earlier slides, the aperiodic FFT plots, like the inset, the C inside the aperiodic activity, so that is differentiated by frequency, right? So
yes,
by frequency it means it's periodic.
No, it's it is frequency dependent, but any source does not have a unique frequency, at least coherently. So if we look at the human data, I think what this really, the way I would interpret this is the aperiodic means they're taking out the big, clearly periodic signals, and they're leaving in the high frequency broadband signals. That's the way I would interpret this. And that's why the blue line is higher, is that across this whole beta gamma distribution, there are a lot of frequency contributors that are not necessarily that oscillatory. They don't repeat their frequency over time. You can also think of it as adding up a bunch of transients, okay, a bunch of changes that are not periodic. They don't repeat. A single change will have a power spectrum, but it will be relatively flat.
Okay, I think I need to work on it to understand better. It's not intuitive to me, as I think.
Well, this is what
where it is.
It'd be good to look at their analytic scripts, how they remove the periodic signal.
That's right.
That'll give you a sense of what it is. I imagine this.
Yeah, I agree.
Cross correlation or regression of periodic waves at all frequencies or something. But who knows?
Well, no, I kind of share your confusion, even though I'm trying to explain it, Ishwara. I have a similar kind of question. In other words, to me, it looks like we should be able to characterize broadband gamma activity and get these same results, and not use their so-called aperiodic measure. Well, try to pick this up in future seminars. My goal is maybe once a month to have a kind of basic science seminar where we go through some of these techniques and talk about next generation things we want to develop.